Okay, hello everyone. Welcome to Penn State CTISI's BERD Seminar Series. My name is Lan Kong. I'm one of the co-hosts for this seminar series, along with my colleague, Dr. Terry Murphy from the Department of Public Health Science. So we have brought you 7 excellent seminars this fall. So today, Dr. Walter is going to present analytical techniques for RNA sequencing. If you are interested in the past seminars, you actually can go to the CTSI website, where you can find the recordings for the past seminars. So this Zoom session is being recorded automatically. If you have any questions, you can tap into the chat or save your question towards the end when we have a Q&A session. Please mute your microphone during the presentation. After the seminar, you will receive the slides recording link and a short survey. We appreciate your feedback about our seminar series. So please complete the survey to help us further improve our seminar series. So now it's my pleasure to introduce today's speaker, Dr. Vonn Walter. Dr. Walter is associate professor in the Department of Public Health Science with a joint appointment in molecular and precision medicine. Since 2019, he has served as a co-director of the Biostatistics and Bioinformatics Shared Resources at the Penn State Cancer Institute. Dr. Walter's primary research focused on cancer genomics. His work has provided significant insights into tumor cell of origin, immune signaling, and molecular drivers of cancer progression. He has also developed several software packages for genomic data analysis. Dr. Walter collaborates extensively across the clinical, molecular, and population science domains. He was a member of the outstanding collaborative research team recognized at the College of Medicine in both 2020 and 2024. In addition, he's an exceptional educator and mentor. He has received multiple teaching awards and guided numerous graduate and medical students. So we are very happy to have Dr. Walter join us today to present an overview of RNA sequencing analysis techniques. So Dr. Walter, thank you for joining us. I will now turn it over to you.
Thank you for the kind introduction, Vonn. And thanks everyone for attending. For those of you that were planning to attend the seminar last week and had it canceled at the last minute, this is the reason why I wasn't able to present the seminar. My daughter came upstairs right before going to school and she said, "Dad, is there supposed to be a big puddle of water on the floor?" And I immediately ran downstairs and I knew there was trouble. So this thing that the red arrow is pointing to is the pressure relief valve, and it was leaking. And fortunately that's been fixed, but we had to have some plumbing repairs in between now. All right, so I want to give a brief outline of the talk. I'm going to start by giving some background on RNA sequencing. This may be familiar to some members of the audience, and if so, I apologize. If not, hopefully this will be of information to you. If you'll pardon the pun, the bulk of my talk will be focused on bulk RNA sequencing analyses because this is where most of my work is. There are lots of different things you can do with RNA sequencing data. I'm going to primarily focus on abundance-based analyses, including but not restricted to things like differential expression analyses and downstream analyses, and also various sorts of clustering analyses. Clustering seems like a natural endpoint for the bulk on our RNA-seq portion of the talk because that gives us a lead into the single-cell RNA sequencing portion of the talk. I don't have extensive experience with single-cell RNA-seq. I have analyzed a grand total of 3 data sets. But it's pretty fascinating stuff, and I have my own perspectives based on this limited work so far, and so I'll tell you my thoughts on bulk RNA-seq versus single-cell RNA-seq, and then I'll talk some more about clustering there. Okay, so background. I love Wikipedia. I support it financially every year, and I would encourage you to do the same. So according to Wikipedia, RNA sequencing is a technique to identify and quantify RNA molecules in a biological sample. And so this cartoon gives you a layout of how a typical RNA sequencing experiment works. You start off with your sample of interest. You extract RNA from that sample and do some sort of target enrichment. Personally, I'm usually interested in messenger RNA, and so we would do poly-A enrichment here. Then the RNA molecules are fragmented, reverse transcribed, the adapters are ligated, and then those are amplified, and so this is the material that is fed into the sequencer. What comes out of the sequencer are sequencing reads, which are just long strings of A's, T's, G's, and C's. The sequences for the adapters is removed. And then because you had reverse transcription to get cDNA, these molecules should be able to match up to the reference genome. And so then you have the alignment portion where you figure out where each of the reads align to the genome. And then you have transcript quantification where you add up the number of reads. And then finally, there are a variety of different sorts of data analyses that can be performed. This is where our focus is going to be for today's talk. I'm not saying that the other stuff isn't interesting, but this is just where we're going to focus on. I do have a tiny bit of experience with step number 5. Traditionally, alignment and transcript quantification were two separate steps. You had an alignment like algorithm like BWA, and that aligned the reads to the genome, and then you had a separate bioinformatics tool which counted the reads up to give you gene level quantification. In the relatively recent past, there have been the development of tools like Salmon, which simultaneously do alignment and quantification all at the same step. The people who develop Salmon are pretty smart individuals, and so there are really good statistical reasons why you want to do that, and it also is extremely fast, so it's great from a computation. As far as I'm concerned, these other steps, they're pretty much magic. It's kind of like asking me, how does my car run? I don't really know the answer. I just know that it gets me from point A to point B. There's a lot of, I'm sure, very sophisticated chemistry and engineering and bioinformatics that's involved in these steps, but that's not what I'm going to talk about. So for us, since I'm interested in abundance-based analyses, things are going to start at transcript quantification. So I'm going to assume that I already have gene level measurements of the data, right? And so from that, I'll talk about various sorts of analyses. As I mentioned previously, I'll talk about differential expression and downstream analyses, also certain sorts of network analyses, such as co-expression, but also regulons, and then finally, the identification of. There are a variety of different high throughput technologies out there. Fill in the name, and you can have some kind of sequencing going on. And a lot of those can be analyzed jointly with RNA sequencing data. What the figure shows here are a tool that was developed to jointly analyze RNA sequencing data and ChIP-seq data. I am not familiar with this tool, but I'm assuming that by jointly analyzing the data, you have more power to detect certain sorts of relationships than you would if you analyzed the RNA sequencing data by itself and the ChIP-seq data by itself, and then somehow you try to merge the results. And so if that sort of thing is of interest to you, there are a variety of different sorts of pipelines out there. I have a bit more experience working with these sorts of integrated analyses. This is an annotation style heat map that I made for Shamara Lawrence's paper that was published earlier this year. There's a variety of different data types shown here, some of which comes from RNA sequencing, some of which comes from other sorts of genomic technologies, and I'll talk a bit more about this sort of thing later. But what we're interested in when we analyze these sorts of figures are co-occurrences, and then trying to use that to motivate some sort of biological story. There are a bunch of RNA-based analyses that I'm not going to discuss. So in particular, detection of gene fusions or alternative splices, or alternative splicing, I should say. One can also do mutation calling using RNA sequencing data and also DNA copy number analysis. my opinion, although I don't have a lot of experience with this sort of thing, RNA sequencing is ideal for analyzing gene fusions and alternative splicing because these are RNA events. And so when you use RNA, when you have RNA sequencing data, you're analyzing, well, the reverse complement, but I mean, the RNA molecules are being directly analyzed. On the flip side, mutation calling, although in theory you can perform it using RNA sequencing data, I mean, mutations happen in DNA. And so if you're analyzing the RNA data, you're analyzing the transcript, and there may be various mutations which occur which are not present in the transcript. You have all sorts of processing that goes on in terms of the transcription machinery going from DNA to RNA. And if your mutations are occurring in there, some of that information might be lost. And then, of course, you have all the information about, or all the issues about different levels of gene expression. And so that may make it difficult to identify some of your mutations if they're in genes that aren't expressed. DNA copy number analyses are close to my heart because that's what my PhD thesis was on. I'm assuming that RNA-based DNA copy number analyses are sort of predicated on the idea that if you have increased copies of a gene, then you're going to have increased copies of the transcript. And so then if you run that in reverse, then if you have increased or decreased levels of gene expression, then perhaps those come from increased or decreased DNA copy number in the genome. But having done some joint analyses of copy number data and gene expression data, the relationship is not nearly as strong as you might expect, and it really varies a lot from gene to gene. And so I personally would have some pretty sincere doubts about someone who's, saying that they're able to identify where a DNA copy number change starts and ends just by analyzing the genes. Anyway, that's just my personal biased perspective. Okay, so bulk RNA-seq. So like I said, there's this particular step in the cartoon that we described previously where you have the alignment of the RNA reads to the genome, and then you have the quantification. And so this is where the data is going to come for us. And so in terms of what does the data look like, you could think about it as a tab delimited text file or an Excel spreadsheet or something like that, where the genes appears in the rows, the samples appear in the columns, and then the entries are the gene level quantification measurements of the expression. So for example, the read count, how many reads align to a particular gene. So in terms of RNA sequencing data, it can have several different formats. The raw sequencing data is in FASTQ format, and each of these little colored rectangles represents a sequencing read. So it's just a sequence of A's, T's, G's, and C's. During the alignment process, the sequencing reads are aligned to the genome based on reverse complementarity. So the sequence in one particular read matches the sequence in the reference genome. And so then the blue sequences align over here, the orange sequences align here, and the green sequences align. And then if you know where the gene boundaries are, then you can just count them up, right? So if the gene A is over here, then there's 6 reads aligning there, and so I have 6 read counts for gene A. If gene B is on the right-hand side of this portion of the reference genome, I have 4 reads over here, and so I have 4 reads for gene. But these gene level read counts can be influenced by a lot of different things. So you can imagine 2 different genes, gene 1 and gene 2, each of which are expressed at the same level. And what I mean by expressed at the same level is that they produce the same number of RNA molecules. If gene 1 is longer than gene 2, then even though the number of RNA molecules that they produce is the same, there's more real estate for those reads to align to gene 1 than there is to gene 2. And so you might naturally expect to have more sequencing reads for gene 1 and gene 2 than gene 2, even though the expression levels are the same. The sequencing depth can also impact the number of reads that align to a given gene. So what the figure here shows is 2 genes, gene 1 and gene 2, which have the same expression level in condition A. And then the same genes, gene 1 and gene 2, have the same expression level in condition B, but they have the higher expression in condition B than they do in condition A. And so naively, you might expect that there would be more sequencing reads aligning in condition B than condition A. But what happens if you have more sequencing reads overall for the condition A samples, just because you have greater sequencing depth there? So suppose you have 3 times as much sequencing depth in condition A than you do in condition B, then you have 3 times as many reads running around. And so even though the genes are expressed at a higher level in condition B, because of the presence of more reads, because of having higher library size, you would get more reads aligning to the genes in condition A than condition B, even though the expression is higher. And then finally, as you might hope, the expression level can also affect the number of reads that align to a given region. So this is sort of a variation of the figure that we just talked about. Here we have genes 1, 2, and 3 in condition A. They all have exactly the same expression level. And so you see the same number of sequencing reads aligning to each of them. Whereas over in condition B, gene number 3 is expressed at twice the level of genes number 1 and 2. And so you see that it has twice as many that align. And so the upshot is that RNA sequencing, or the thing that's important to keep in mind, is that RNA sequencing measures relative expression. It doesn't measure absolute expression. And because you're measuring relative expression, there are lots of things that you have to account for when you're trying to quantify expression measurements. Okay, so as I mentioned previously, the gene length, the sequencing depth, and so on. And so this has led to a veritable alphabet soup of different ways to quantify gene expression. So you can have CPM, counts per million map reads, TPM, transcript per million map reads, and so on and so forth. And so each of these different measures has their benefits and liabilities. I strongly encourage you to take a look at this wonderfully titled article, What the FPKM, which was, admittedly it's over 10 years old, but it's a great read, and it really illustrates some of the things that you need to think about when you're quantifying gene expression from RNA. Okay, so we have this abundance-based analysis that we're going to do. We're starting off with our gene level read counts. Before we begin to work into the analysis itself, there are various pre-processing steps that we want to do. There are certainly going to be genes that are lowly expressed or perhaps not expressed at all, so you don't have any read counts for those genes. We're going to do some sort of multiple testing correction because we're looking at genes across the genome. And so we want to filter out those lowly expressed genes in part to reduce our multiple correction penalty. As mentioned on the previous slide, we definitely want to control for differences in library size. And there may be additional sorts of normalization steps that we want to do. So if we're combining data from different experiments, then perhaps we need to think about some sort of batch correction. There are a variety of different tools that are available to do this sort of thing. I am personally a really big fan of this R package called RUV-seq for removing unwanted variation. It uses housekeeping genes to do normalization procedure. And in my experience, it works really, really well. Finally, we might be interested in doing some sort of exploratory analyses just to try to get a look at the data, whether that be principal components analyses, unsupervised clustering, and so on and so forth. So in terms of differential expression, so I'm gonna make this overly simplistic here. Often we're interested in just comparing two groups. And so the naive thought is that, well, if all you're doing is comparing experimental group versus control group, then you're just going to do 20,000 two-sample t-tests. Now, we all know that it's not really quite this simple. And there are various complications associated with this. For example, because you're doing so many tests, you want to do some sort of multiple testing correction. But perhaps, and equally importantly, you want to ask yourself if the assumptions are satisfied. It's probably a pretty safe bet that if you're analyzing the read counts themselves, the assumptions for a two-sample t-test are not satisfied. Two-sample t-test will not allow you to control for relevant variables such as batch, and it won't enable you to deal with more complicated experimental designs. And so that's probably not the approach that we want to take. The most commonly used approaches are based on linear models. So the three that I mentioned here, lemma plus the Voom transformation, DE-seq2, and EDGE-R, these tools have been around for a long time, but they really do seem to be the industry standard. I'm not saying that there isn't room for improvement over them, but they're just very widely used. Lemma is a tool that's been around since the days of microarrays, and then the Voom transformation was introduced in 2014. So basically you apply the Voom transformation to your read counts, and then you can feed that data into Lemma and do your standard linear modeling. DE-seq2 and EDGE-R are slightly different. They use generalized linear models, so then you're analyzing the read counts directly, but you're fitting a negative binomial regression model. And then you can do the standard linear modeling or generalized linear modeling approaches. Importantly, you don't want to use counts per million or transcript per million or FPKM, et cetera, in your statistical analyses. All of these tools are specifically designed to analyze the read counts directly. What's unfortunate is that a lot of gene expression data is publicly available and it's in these formats. Okay? And so if you have these formats and you have the read counts available, that's great. Use the read counts. You can use these sorts of things for various exploratory analyses, but if at all possible, you don't want to do differential expression using it because it's just the inference is not as robust. Because all of these approaches are based on linear modeling techniques, they allow you to consider more complicated experimental design, so sort of matching design. They'll also allow you to correct for batch as a covariate, and that is the preferred way to do it. You want to include, if you have some sort of batch issue that you've identified in your exploratory analysis, you want to include batch as a covariate in your design matrix, as opposed to doing batch correction and analyzing the corrected reads. These methods also are fairly sophisticated and they give you better estimation of the gene-wise variances, and that in turn leads to better. The output of these algorithms includes various things, such as some sort of test statistic. So basically what's going on is you're doing a hypothesis test at the gene level. So each gene is going to give you a test statistic. You're going to get a measure of fold change, which is how much did the expression change, and then you're going to have some sort of measure of significance. And the fold changes and the measures of significance are typically illustrated in a volcano plot like this. So on the x-axis, you have the log fold change. The upregulated genes appear on the right. The downregulated genes appear on the left. And then you can have various significance thresholds for the fold change and significance threshold for the measure of statistical significance. You may also choose to visualize some of your favorite genes in a heat map, and there you're just visually representing what the expression measures are. Now, a differential expression analysis may identify hundreds or thousands of differentially expressed genes, and you want to try to make sense of them, i.e. to try to turn that information into some sort of biological story. And so that leads to your downstream pathway or enrichment analyses. And so basically the question that you're asking when you're doing this sort of pathway analysis is my biological pathway of interest, or is my favorite gene set enriched for my differentially expressed genes? I am personally a big fan of gene set enrichment analysis pre-ranked, GSEA pre-ranked analyses, and I'll describe those in a second. Go analyses for gene ontology analyses are also popular, and there's a whole laundry list of possible. The way a GSEA pre-ranked analysis works is you start with an ordered set of genes and a gene set of interest. So the ordered set of genes comes from your differential expression analysis, and you can order them perhaps according to the log fold change, or perhaps according to the test statistic or something like that. And you want to order them in such a way so that the most upregulated genes are at the top of your list and the most downregulated genes are at the bottom. And then the gene sets are going to come from some sort of curated database. This is a screenshot of MSigDB, which has lots and lots of different gene sets, including the cancer hallmark gene sets corresponding to some of the hallmarks of cancer that were studied by Hanahan and Weinberg. And so two of those hallmark gene sets are, for example, the interferon gamma response gene set, or the hallmark G2M checkpoint gene set, which is genes that are involved in regulation of the cell cycle. The way the gene set and the GSEA pre-ranked analysis works is you have some sort of running sum statistic that you calculate as you walk through the gene list. So you start at the top of the gene list, which is your most upregulated genes, and you work your way down to the bottom of your gene list, which is the most downregulated genes. And so your walk starts at 0. And then as you walk through the gene set, you either take a step up if you encounter a gene in your gene set, or you take a step down if you encounter a gene that's in the gene universe, i.e. all the genes that you analyze, but is not in your gene set. Now, the size of your gene universe is much bigger than the size of your gene set. And so you have a lot more down steps than you do up steps, but the up steps are a lot bigger. And then because of the size of the steps, if your walk starts at 0, then it must end at 0 as well. And this leads to the sort of GSEA plots, which look something like this. So on the bottom here, basically what I have is the genes appearing in ranked order. And every time I have a vertical black line, that corresponds to a gene in my gene set. So I have a whole bunch of black lines that are on the left-hand side, which is where the walk starts. These are the most upregulated genes. And so every time I encounter one of these black lines, I take a whole bunch of steps up to this peak, and then I'm pretty much done with all the genes in my gene set. And so I keep taking a whole bunch of steps down until I get to 0. The second GSEA plot shows the reverse scenario, where most of the genes in my gene set are at the bottom of my list. So these genes are downregulated. I started my walk at 0, and then I take a whole bunch of down steps until I start encountering genes in my gene set, and then I start walking back up to 0. And then finally, if my genes are just sort of randomly distributed throughout, the genes in my gene set are randomly distributed throughout the ranked list, then sometimes I go up and sometimes I go down, but it's really just, there's no real pattern. And what you're interested in when you create this running sum statistic is you're interested in the maximum deviation from 0. So that's your test statistic. And then you can compute some sort of p-value based using a permutation-based approach. And then usually you're analyzing multiple gene sets, not just a single gene set. And so you have to do some sort of correction for multiple. Go analyses are slightly different. Basically, for each gene, you ask yourself, is it differentially expressed? Yes or no. And is it in my gene ontology term of interest? Yes or no. And then you just count the number of genes that appear in the various cells, and then you do some sort of Fisher's exact test. And then based on the p-value that you get from Fisher's exact test, you'll want to do some sort of correction for multiple testing, and then you can display the results in this sort of format. One of the reasons why I prefer a GSEA pre-ranked analysis is that it has some directionality associated with it. You can have upregulated and downregulated, and you can get a sense of whether there's a difference there. Whereas in your typical Go analysis, although I guess you could compute you could do a one-sided Fisher's exact test, typically that's not done. Now, I don't know if this is a common experiment, but it's not an uncommon experiment to have data from 3 control cell lines and 3 experimental cell lines. You do your differential expression analysis, and then you follow it up with GSEA, pre-ranked, or Go. And, you know, there are some potential issues with doing this sort of analysis. So Go terms are biased towards well-studied genes, and I'm sure you could say the same thing about the gene sets that you're going to find in your curated gene sets. Multiple testing is a problem, and these things probably aren't independent because there are lots and lots of Go terms, and they have lots of genes in common. So, you know, if you're doing a false discovery rate correction, that may not be 100% appropriate. You have, of course, the inevitable issue of correlation versus causation. We're thinking about it in terms of causation, but it may just be correlation, and so on and so forth. All of these points, I think, are valued or reasonable arguments to raise when somebody is presenting results from the sort of pathway analysis. They're not perfect, but at the end of the day, we're just trying to provide some sort of insight into our results. And so that's why they're commonly used, and that's why I can. Other abundance-based analyses, you can do a single sample GSEA. So here, basically what you do is you think about the ordering of a gene in the genes in any particular sample. And so there's no sort of testing going on. You're just looking at the actual expression measurements. If you think about the GSEA walk that we described previously, there's nothing to prevent you from doing that exact same walk for each sample separately. And then that enables you to compute this sort of enrichment statistic on a sample by sample level. This is the sort of thing we did in this figure that I mentioned previously from Shamara Lawrence's paper, this annotation track corresponding to the interferon gamma pathway. This was a single sample GSEA analysis using the interferon gamma genes from MSigDB. And what you might notice is that the scores are very red over here. That means they're high in this basal squamous gene expression subtype. If you do a Kruskal-Wallis test, you can verify that statistically significantly. And so that makes sense because of what we know from the underlying tumor biology. Regulon analyses are similar. Regulon analyses briefly is a machine learning approach which enables you to explore expression patterns of genes that are co-regulated by a transcription factor. So STAT1 and FOXA1, those are both transcription factors. And then what the machine learning algorithm does is it analyzes all the gene expression data and it identifies genes that have expression patterns that are sort of similar to what you would see in the expression patterns for these particular transcription factors. And so one nice thing about Regulon analysis as opposed to the analysis that I just described is that the regulons, which are the genes that are regulated by the transcription factors, that's learned from the data. It's not, so it's more of an unsupervised approach, whereas when I'm doing my single sample GSEA analysis, I have to pick out the gene set. So that's more of a super. Finally, you can do various sorts of principal components and supervised clustering analyses. You can do these for different ends. You might do them initially when you're trying to get a sense of your data, so it's some sort of quality control or data exploration analysis, but you can also do it to try to identify molecular subtypes, and I'll talk a bit about both of them. So here I have a principal components analysis plot and an unsupervised gene expression heat map from an analysis that I did for Josh Warwick. Basically, this experiment involved injecting bladder cancer cell lines into mouse flanks and then watching the tumors as they grow. So here, we were interested in looking at the gene expression data because we had a parallel experiment where we measured tumor volume longitudinally for mice that were subjected to various sorts of treatments and led to different rates of tumor growth. And so the RNA-seq analysis was conducted to try to get some sort of biological insight into what was causing these differences in tumor growth. And so I have 6 different treatment groups. The PCA plot is nice because you can see that all of the points that are the same color, which are biological replicates, they all seem to cluster together. You see that the olive points cluster together with the green points. The olive points are points that corresponded to knockdown of JAK1 and JAK2, and they cluster together with the olive points are the JAK1 knockdowns, and they cluster together with the green points, which are the JAK1, JAK2 knockdowns. And so because those are so similar, that's suggesting that JAK2 knockdown doesn't have very much of an effect in this. And that's sort of reiterated over here, where you have the scramble, which is our control. They seem to cluster nicely with the JAK2. And then that's visualized over here in the gene expression heat map. Most of your biological replicates cluster together and exhibit similar sorts of expression patterns. And so, you know, these are the sorts of things that as an analyst, I like to see, right? So I don't have any weird outliers. The data is behaving the way that I want it to, and so that I can move forward with my differential expression analysis, see what it has to do. Molecular subtypes arise typically in larger genomic profiling studies. This is the graphical abstract from the 2017 paper by the Cancer Genome Atlas, where they were exploring muscle-invasive bladder cancer. And so here you can see that the microRNA subtypes are front and center. Molecular subtypes first came onto the scene in the early 2000s. This is a figure from the 2001 paper by Sorley that was one of the first papers to ever introduce breast cancer gene expression subtypes. On the left-hand side, you have a heat map of 456 genes and 85 samples, and what the various colored lines represent here are groups of genes of interest, and then what you have on the right is sort of a zoomed in version of those particular portions of the heat map, and you're exhibiting high expression or low expression of some of those genes. If you look more closely at the dendrogram, you can see that a total of 6 gene expression subtypes were identified, and these have been recapitulated over and over again in molecular studies of breast cancer. And one of the reasons why this paper has 14,000 citations is because the gene expression subtypes that were identified were found to be strongly linked with outcome. So the basal-like breast cancers, they have very poor outcomes. The luminal A's have very, very good outcomes. And so this was quite remarkable discovery at the time because, you know, molecular profiling techniques weren't widely used. It was also found that the gene expression subtypes did a better job of predicting survival outcomes than a lot of the clinical markers at the time. And so this really opened the door for molecular profiling studies to, you know, to show their power. Now, a reasonable question to answer when you look at this dendrogram is, why are there 6 clusters? And the reasonable answer is, well, because they colored it that way, or because that's where they chose to cut the dendrogram. But typically, when you cut a dendrogram to try to identify clusters, you cut it the other way. Okay? You don't cut it parallel to the dendrogram, you cut it perpendicular to the dendrogram. And so if you cut the dendrogram here, you see that it doesn't separate the red group from the pink group. Those are still in the same branch of the dendrogram, whereas at least you're able to get the, you know, the normal X, luminal C, luminal B, and luminal A. If you want to bring this cut point down a little bit to separate the red group from the pink group, then you end up separating something else too. And so this begs the obvious question, which is, did they do some sort of manual adjustment to figure out where they wanted to cut the dendrogram to identify their molecular subtypes? And I would argue that the answer is yes. And that's fine, because obviously the subtypes that they identified had clinical importance. But the point that I'm trying to make here is that there might be a little wiggle room here in terms of identifying your groups when it comes to this clustering analysis. There are a variety of different approaches that you can take for clustering analyses. You can have connectivity-based approaches, then that leads to some sort of hierarchy of clusters that you visualize in a dendrogram, as opposed to partitioning-based approaches. I don't want to get too deep into the weeds here, but a key difference between the connectivity-based approaches and the partitioning-based approaches is that when you do a connectivity-based approach, the number of clusters is not chosen in advance. Okay, you look at your dendrogram, you might look at a heat map, and then you try to get a sense of, okay, there are this many clusters, there are 5 clusters, there are 4 clusters, or whatever. Partitioning-based clustering is different. You pick the number of clusters in advance, and then you find the objects that cluster together. And so, personally, basically all my experience in this sort of area has been in applying agglomerative hierarchical clustering. And so here the goal is we start off where every subject is its own cluster, and then we combine them to get groups of objects which are all similar to each other. So the underlying premise behind clustering is that the objects in the same cluster should be similar to each other. And this brings up the obvious question, well, how am I going to measure similarity of objects? And then because objects get grouped together to make clusters, you have to ask yourself, how am I going to make similarity? How am I going to measure similarity of clusters? Our objects, they're just measures of gene expression. So we can think of X as an object, and X1 up to Xn are the gene expression measurements for that particular subject. Y is a subject, and Y1 up to Yn are the gene expression measurements for it. And then there are various mathematical formulae that you could apply to measure the distance between these two things. And I'll let you think about those. In the interest of time, I'll move on. Once you know how to measure distance between objects, then you have to start to think about measuring distance between clusters. And this is the standard sort of mathematical trick. You give me a new problem to solve, and I try to reduce it to a problem that I already know how to solve. So I'm going to define the distance between clusters based on the distance between the objects which are in the clusters. And so this is called linkage. Single linkage, the distance between two clusters X and Y is the smallest distance between any objects in X, object little x in X and little y in Y. Complete linkage is sort of the opposite. The distance between any two clusters is the maximum of these distances between objects, and average linkage, as the name implies, is the average of those sorts of things. The linkage that you choose will have an effect on the shape of the clusters that you get. Single linkage can tend to lead to clusters which are kind of long and strung out, whereas complete linkage and average linkage give you more sort of glob-like clusters, which is personally what I'm interested in finding. Previously, I mentioned the TCGA bladder cancer paper from 2017. That paper identified a total of 5 gene expression subtypes. And what this figure shows is expression patterns of various genes in the different subtypes. And so this is all well and good, but I was curious to see how were the 5 clusters identified. And so if you look in the supplement, it says that the expression clustering analysis was done by combining 2 different approaches: Bayes non-negative matrix factorization, which is one clustering approach, with consensus hierarchical clustering. And so somehow they took these 2 clustering approaches and pulled them together to identify their clustering solution. And if you look a little bit further down in the same passage, it says that 9 out of 20 independent Bayes NMF runs gave you a 5-cluster solution, while 11 runs gave you a 4-cluster solution. And then after some sort of manual inspection, they decided to end up with a 5-cluster solution. So this, I bring this to light because it's very similar to what we saw previously with the Sorley paper. How do you determine the appropriate number of clusters? You know, that one sort of approach might lead you to one number, one sort of approach might lead you to another number. At the end of the day, you have to make some sort of executive decision, and your choice might actually be made by which enables you to tell the best story, right? And that's okay, because as my thesis co-advisor used to say, clustering is just exploratory data analysis. And so if one clustering solution gives you a better biological story than another clustering solution, then by all means, you should go. All right. Finally, moving on to single-cell RNA-seq. So having discussed a bunch of bulk RNA-seq analysis approaches, a reasonable question to ask is, which of them carry over to single-cell RNA-seq? As I said, I've only analyzed a total of 3 single-cell RNA-seq data sets, so I won't pretend to be the authority, but it seems like most of the analysis approaches carry over. You can do differential expression analysis, you can do pathway analysis, you can do all these other sorts of things, all right? But I think what's important to understand is that when you're doing single-cell RNA sequencing, you're asking a different biological question. So if we go back to this example that I mentioned previously, the tumors that were obtained from these mice in the 6 different treatments, remember that we had done this parallel experiment where the mice, where we followed the longitudinal tumor volume for mice subject to different treatment groups. And so we saw that different treatment groups led to different rates of tumor growth, and we're trying to get an insight into the underlying biology. So that led to the selection of these different experimental groups in the RNA-seq data. And so basically the question that we're asking is, how do the different treatments change the gene expression patterns? And so if we know how the treatments change the gene expression patterns, maybe that will give us insight into the changes in tumor volume as well. On the flip side, here's a project that I was involved, a single-cell RNA sequencing project that I was involved in. And so in terms of the experimental design, there were 4 possible conditions. So I had PD1, the gene, I either had wild type or knockout, and then I had cells which were coming from either CD4 or CD8. And I had data from a total of 16,000 cells, as opposed to the bulk RNA-seq analysis that I just described, where I had data from a total of 18 mice. So the question that we're interested in this single-cell RNA-seq analysis is, does the PD1 knockout change the immune cell population, and are these changes different in CD4 and CD8 cells? And so if you think about it, the question that we're asking is, how many different populations of cells are there? And then what characterizes these different subpopulations of cells? And so if we're trying to figure out subpopulations of cells, those are going to be groups of cells that have similar expression patterns. That's clustering, right? So if we're trying to figure out how many subpopulations of cells, we're going to do some sort of unsupervised clustering analysis. And then if we're going to try to figure out what characterizes these different populations, that's your standard sort of RNA-seq analyses that we've been talking about previously, like differential expression or whatever. Now, in terms of the actual data itself, it's pretty different between bulk RNA sequencing and single RNA sequencing. With bulk RNA-seq, your sample numbers are in the tens, maybe up into the hundreds. In the clustering analysis you do, you're going to restrict attention to, say, the 1,000 to 2,000 most highly expressed and highly variably expressed genes, because those are the genes that, in some sense, are doing the talking. And then you cluster. In terms of the number of cells that you're looking at in a single cell RNA-seq analysis, it's literally orders of magnitude larger. I mean, you're going to have cell numbers in the thousands to tens of thousands. You're going to, again, want to restrict attention to highly variably expressed genes, because those are going to be the genes that show your difference in expression patterns, but the data is just too big. I mean, you can't analyze 15,000 cells and 2,000 genes. It's just going to be too large of a data problem. And so, unlike bulk RNA-seq, with single RNA-seq, we do a dimensionality reduction step. And so the clustering analysis is performed in PC space, not in the original gene space. And there are different sort of dimensionality reduction techniques that can be applied. Principal components analysis is a linear dimension reduction. The math is easier over here, and so I'm guessing that's why that's where the clustering is done. You have these non-linear dimension reduction techniques that lead to these sort of really beautiful UMAP plots that are great for visualizing the clusters that you identify, but it's, I think, that just in terms of the computation is just way too difficult to do in that non-linear space. The tool that I've used in my single cell analyses has been the Seurat R package. It's an amazing suite of tools. I can't recommend it highly enough. It uses what's called Louvain clustering, or at least that's the default, which is a graph-based approach. And so each of your cells in your experiment correspond to nodes in the graph, and those are aggregated into shared nearest neighbor communities, and those are effectively clustered. And so the thing that controls the number of clusters is this parameter, which is called the resolution. Different values of resolution will give you different clustering solutions. And so this is the same single cell RNA-seq data that I mentioned previously. Here's a 6 cluster solution, a 7 cluster solution, a 10 cluster solution, and a 11 cluster solution. And so how do we figure out which is the best clustering solution, which one is optimal? And as you may have guessed, there might be some subjectivity involved, and again, I think that that's okay. One of the things that you can use to get a sense of what is the optimal clustering solution is this figure over on the right, which is called a silhouette plot. And so the way a silhouette plot is organized is you have your cells appear along the x-axis, and then the y-axis measures what's called the silhouette score. And so for each particular cell, you have the silhouette score, which is a measure of how similar is it to members of its own cluster versus how similar is it to members of different clusters. And then the cells are ordered in decreasing silhouette width for each cluster. Cells corresponding to positive silhouette widths are more like their own cluster than other clusters. And so you could reasonably argue that, well, that means that they're probably in the right place, right? If they're more like members of their own cluster, then they were correctly clustered. Whereas cells which have negative silhouette widths, those are more like different clusters than they are members of their own cluster. And so you could argue that they were misclassified. Naively, you could say then, okay, well, I want to find a clustering solution that gives me only positive silhouette widths. And that's a reasonable goal, but if you take it to its illogical extremes, if you think about the possibility of having only a single cluster, then there's no other cluster to belong to. And so if there's only one cluster, all cells will have positive silhouette widths. And then if you take it to the other extreme, each cell consists of its own cluster. And so no cell can be more like anything else other than itself. And so that's another situation where you're going to have only positive silhouette widths. So negative silhouette widths are probably going to be along for the ride. You just have to figure out how you want to deal with them. So here's what happens when we increase the resolution a little bit. Before, all of these guys were cluster 0. Now we split it up into a new cluster, cluster 2. Some of these guys in new cluster 2 have negative silhouette widths. You could argue that that is a bad thing. And some of these guys in cluster 1, which used to have positive silhouette widths, now have negative silhouette widths. And that's probably because of the presence of poly. We ended up going with this 11 cluster solution. We went through a variety of different options, including separating or clustering the CD4 and the CD8 cells separately. But we eventually, this was the one that led to the best biological story. In addition to the silhouette plots, one of the things that guided our decision on choosing this particular clustering solution is a heat map of differentially expressed genes. And so we look at different groups of genes here that characterize some of the different clusters. These complement genes characterize cluster 0. These CD8 genes characterize clusters 0, 1, and 3, which makes sense because all of the cells over on the right here are CD8 cells. These CD4 genes characterize clusters 2, 4, 5, and 6. The CD4 is over on the left, and that's where clusters 2, 4, 5, and 6 are located. But then, there was this cluster that was characterized by FOXD3. That was a very interesting to the people that I was working with. Similarly for the cluster with CCF7 and so on and so forth. And so then we summarized all of this information into this GRIDY heat map. Almost done. I want to mention briefly this experiment that I did for Todd Shell. This was literally the first experiment that they ran on the BD Rhapsody instrument that they got earlier this year. There are a lot of technical issues, and so the yield was really small. We only had data for like 1,000 cells, but in spite of only having 1,000 cells, we were able to identify a lot of different clusters. And another really cool thing about Todd's experiment was that his cells were much more heterogeneous. And so I applied a R package, which will classify each particular cell into different sorts of cell types. So there are lots of different immune cells, granulocytes, dendritic cells, monocytes, and so on and so forth. And so the cell type prediction algorithm gave us these cell type predictions, which are shown in the top annotation track. And what was really cool to us is that the clusters that we identified from our clustering solution were very concordant with those cell type prediction labels. And so this is really what you want, right? So the whole idea is to try to identify these sort of groups of cells that have similar expression patterns and then say, what kind of cells are they? In closing, I'd like to thank all my great collaborators, David Graff, Josh Warwick, who unfortunately are no longer here at the institution, and then Aaron Lukacher, Todd Shell, and Ramiza Ali, who helped me with, or who provided single cell RNA sequencing data. And so with that, I'd be happy to answer any questions that you may have.
Thank you. This is a wonderful talk. There's so many information in 1 hour. So any question from the audience? Feel free to speak up. Maybe I'll start with one simple question. So I heard you talk about the batch effect. So it seems like you can incorporate a batch as a covariate in the analysis stage.
Right.
Is there anything that researcher can design a better study to reduce the batch effect when they design the study or when they measure this data?
Yeah, yeah. I mean, certainly there are different things that you can do, for example, in terms of just the processing of it. So I don't know a lot about this, but in terms of the processing of the samples, you don't want to process it in batches, right? So if you have 3 different experimental groups, you don't want to process those samples all on 3 different days, potentially using 3 different sets of reagents, right? Because the different sets of reagents might behave a little bit differently. So the people who run the cores should know about these sorts of things, but I think it's always good to follow up with them to do what you can to prevent the introduction of batch effects.
I see. Thank you.
And the other thing that I would say is that even though there are these tools for removing batch effects, the better approach is just to not to have them in the first place, right? I mean, if you can not have any batch effects, then great, you don't have to control for them, and that just makes your life so much easier. And they're not a magic spell, I mean, you can correct some, but if you correct too much, you run the risk of literally correcting out the biology that you're interested in discovering, and you definitely don't want to do that.
We got one question. It's a comment in the chat.
Okay, I can't read it, sorry. Maybe if I stop sharing.
Yeah, you can stop sharing.
Okay. If you're using any Promega, yeah. So this is I'm not familiar with Promega. There are a lot of sort of ready-built pipelines. For example, the single-cell RNA sequencing tools that process the raw sequencing data. Some of them will do clustering for you, right? Maybe I guess I'd like to think that doing the job after the fact and not just using the one-size-fits-all tools is going to give you better results. Maybe I'm saying that because I want job security, and so I want to be able to continue to analyze this sort of data. But I think that these sort of they're great for what they are, right? I mean, I would never be able to create them, so I don't want to speak poorly of them. But I think if you do the analyses yourself, you just open yourself up to being able to discover a lot more, right? If you limit yourself to the things that the dedicated pipeline can find for you, you're never going to find anything outside of that realm. And I've been doing gene expression profiling for almost 15 years now, and I still learn new stuff all the time. So there's really just a whole universe of tools out there. And so I would encourage you to do as much as you can. Yeah, Terry.
Hi, thank you so much, Vonn, for this great introductory talk. I've spent most of my career modeling clinical outcomes, measured directly from people. So I've always tried to get my head around, like genes are sections of DNA code, and then you're looking at clusters of genes. And I'm wondering, I guess it assumes that the technology has no error, right? That there's no measurement error, right? You kind of take the genes you have. But when you build the clusters, do you ever try to interpret them like genetically or physiologically?
I would say that what I described with the Sorley paper, where they found this association between the gene expression clusters and the patient outcomes, that's the holy grail, right? You can always find patterns in the gene expression data. They're always there. But if it's not meaningful, then it doesn't tell you anything, right? I mean, you would expect to see differences if you get, enough cancer patients together. Some of them are going to have similar gene expression patterns. And so what you really want to do is sort of draw some biological or clinical insight out of that. Like maybe, like one of the things that we did with a project that I was involved in when I was at UNC was we found this is in head and neck cancer, but we found that this group of patients that had similar gene expression patterns, they were highly enriched for being HPV positive. And so these, and HPV-driven head and neck cancer has very different clinical and biological outcome than non-HPV driven cancer. And so that was reflected in the gene expression patterns. And so that's really what you're trying to do. You're just trying to get insight into the disease. That's the way that I think about it.
Sure, sure. And there's a real like probabilistic aspect to this, right? So I guess if you're one study, you identify a cluster of genes and you say, oh, this is typical of HPV, then from then on, other studies who find that, they also look for that same association with HPV, and slowly the science builds on itself like that.
Well, yes, but there is always the issue of reproducibility, right? So your particular cohort may have found such and such, and then somebody else does a study with their cohort, and they don't find exactly the same things that you find. They might find some of the same things, but they might find some different things, right? And so what happens, and you can sort of see this evolution over, the lifetime of gene expression profiling. If you look at bladder cancer, for example, some of the early studies maybe only identified 2 or 3 expression subtypes. Now it's widely recognized that there are at least 6. And so basically they've sort of refined, so the main dichotomy in bladder cancer is luminal versus basal. But what they found is that the luminal can actually be subdivided into several different luminal groups. And so that, if you have sufficiently large cohorts, better data quality, then you can sort of tease these things apart and then, again, get insight into the disease itself.
And looking at the far long term, I imagine the idea is you get so precise, so repeatable, that you can identify clusters of gene expression with a high rate of confidence, so you can develop medications or interventions or gene therapy.
I'd like to put on my salesman hat and tell you that that's actually going to happen. But I think that's, as much as I'd like that to be true, I think it's just too far-fetched. I think about everything through the lens of cancer because that's where most of my experience is. Every tumor is different, like literally every single tumor is different. And so, one, you're not going to see those same. You're not going to have enough knowledge to be able to take an individual's gene expression profile and categorize them somehow in a highly specific and meaningful way. You may be able to categorize them broadly, like, oh, your tumor seems to have a basal gene expression profile, and so therefore maybe these treatments will work better for you than these other sorts of treatments. But I think there's going to be, at the end of the day, there's only so much you can do, right?
Great. Okay, thank you so much.
All right. Thank you everyone for joining us. Our last seminar is scheduled for December 8th, so we hope to see you then. Everybody has a great rest of your day. Thank you. Bye-bye.
Great. Thanks everyone for attending.